All right, thank you so much. I'm actually going to be co-presenting with Lauren SEGARRA, who was a graduate student at my lab, and we're going to be talking about some trait architecture work around drought tolerance in sunflower. And because this is a pollinator conference, we'll be focusing on floral traits. And the reason Lauren will be co-presenting with me is because most of this work is actually her master's thesis. I can't see the chat very well, so if someone tells me if I need to look at that, please do. So this project is a subset of a larger project, which I know quite a few people on this have been involved in some way. It's evolutionary genomics of abiotic stress resistance in wild and cultivated sunflowers. And there's a lot of collaborators. I just listed the people that we worked with most closely here. And I just want to acknowledge Andres Temay right when we start, because he did a lot of work with us on this project. And this large project is looking at a lot of different abiotic stress tolerances in the Sunflower Association mapping population. And that diagram on there is just from this Mandel 2011 paper, and it's showing that whole population, which is about 433 lines. And this project is looking at a 288 genomic diversity panel that captures about 90% of the allelic diversity in this population. So I just wanted to give you the overarching context. So the main objective of the study I'm going to be talking about today is actually not to look at that whole diversity panel. They're doing that through UGA. We at Central Washington University were looking at small subsets of the SAM population in more detail, looking at different abiotic stress tolerances. And this particular talk is looking at trait architecture and its association with drought tolerance in 40 SAM lines, focusing on drought. And like I said before, if you want to find a lot of these results published, there is a reference right there on Lauren's master's thesis. All right, so Lauren, you can go ahead and talk about this slide.
Okay, hi. Can y'all hear me?
Yes.
Okay, cool. All right, so the main issue that we wanted to highlight is drought and water stress, and that it reduces seed yield because sunflower is an important oilseed crop, and the main purpose of a lot of this research is to improve seed yield. So we were looking at floret morphology and nectar as some of the main floral traits that were under our study. So floret size has been shown to be reduced under drought due to high water and carbohydrate costs involved in the process of flowering. And studies have shown that floral nectar production is positively correlated with soil moisture. So this is important because pollinator attraction may influence crop yield. And we wanted to look at this in particular in the field, in situ, and floral traits had not yet been studied in the SAM population in the field. So our experimental design involved two treatments, drought and watered. There were 40 SAM lines that we utilized. I think we had, 640 individuals planted in the field on May 18th, 2019, and 598 total individuals survived to harvest that we were able to collect total data from. So I think we can go to the next slide. The traits measured were size traits such as stem height and diameter during the midpoint and the final harvest, head diameter at flowering, so the total size of the ray flowers. The floral traits we measured were the corolla length, anther length, and style length. The nectar traits we measured were nectar volume per floret and nectar sucrose concentration by volume. And we estimated the apical inflorescence mass, seed total. Well, we estimated the seed total based on a calculation of pretty much doing like the what was that? The diameter of, oh man, the circle. Sorry, y'all, I'm sleep deprived. Okay, we'll move on. So this slide just kind of illustrates some of the field methods. We used a microliter micropipette in order to sample nectar from 10 florets haphazardly at a time, and then put that nectar on a prism refractometer, and we're able to estimate the sucrose concentration after many calculations. We also sampled florets in the male stage and florets in the female stage from each plant individually. Put those, dissected those, put them on a number line. We're able to scan the image and then used ImageJ software in order to do our measurements so that we didn't have to do those measurements physically. We were just measuring from an image that was to scale. So. Next slide. This slide is just looking at the significant differences between the control and the drought, and it shows that a drought effect was observed for all size and yield traits. So yeah, significant drought effect. Seed total was smaller in the drought experiment than in the watered experiment, and other size traits were smaller than in the watered, as expected. Okay, next slide. And then of the floral traits, we observed that head diameter, corolla length, and style length, and nectar volume per floret were all had significant drought effects. However, the only one that did not necessarily have a drought effect was anther length and nectar concentration by sucrose concentration by volume, which is the NCV. So we can move to the next slide. Yeah. Okay, and then these diagrams show significant line by treatment effects for some traits. So for example, we have head diameter at time of flowering. These are just showing line effects, pretty much like how lines behaved in the drought treatments versus the water treatments. And you can just see that it pretty much just illustrates a lot of genetic variation, but also illustrates drought effects in a different way.
Okay, I think this is me, right?
Yeah.
So just to add on to, this is actually a good time to say this again. So what Lauren was saying, so the way that we estimated seed total in this particular experiment was by taking the diameter, the number of seeds along the diameter of the apical inflorescence, and then calculating an estimated seed total based on that. Instead of cleaning heads in this particular experiment, I have done that as well. Sometimes, you know, there's only time to do certain things. So this particular graph is looking at what we did here is took estimated seed count by line, and those numbers on there are the different lines in the water treatment, which is down here on the x-axis, and then estimated seed count by line in the drought treatment, and did a regression of those two. And this big black line in the middle here is the 1-to-1. So if the plant produced the same amount of seed under watered and drought, it would be on that line. So not surprisingly, the regression is below that line because you would expect that plants are producing genotypes on average are producing fewer seeds in the drought than in the water treatment. And what this is it's one way to estimate drought tolerance in this population. So what we're looking at here is if you look at the population as a whole, you can argue that the plants that are above that line are more tolerant to the stress, and ones that are below the line are less tolerant to the stress or more susceptible to the stress. And we looked at this actually in 2 years. This particular graph is showing only 1 year, and found 5 of the SAM lines that we looked at that we identified as the most tolerant from this graph. And then another 5 of the lines that we identified as the most susceptible. And these 2 little stars here, so only 2 of these lines were in the top 10, were estimated in the top 10 most tolerant to drought when we did this experiment, actually over 2 years. And none of the most susceptible lines came up in both years. So those were changing a lot. There's a lot of environmental effect on this, basically. So we also looked at a different way to measure intolerance, and I wanted to cite Andres Temei here because this is a paper that just came out in 2020, and he was arguing to look at tolerance in another way, and that's this expectation deviation tolerance. So this is a little bit different version of that first graph, and what you're looking at here is the performance of a line in the benign, in this case would be watered conditions, versus the change in its performance. So what you're looking at here is basically how good lines are at maintaining performance under an abiotic stress. So we also looked at that in our study and found actually pretty similar pattern to what a lot of other people have found in these lines, that plants that had high yields when they were watered also had higher reductions in seed yield when stressed. But this pattern actually isn't super strong in our study. We looked at this in 2008, and I don't have the graph here, but the graph is much stronger in 2008. In this study, this 2019 one, we do see that pattern, but it's not super strong. And it's probably driven by some lines that actually produce more seed under the drought or water stress conditions than the watered conditions. This is just showing a couple of the traits and their correlations with tolerance. So the next step is to start looking at which traits are associated with these different tolerance measures. This particular one is looking at the first measure of tolerance, not the expectation deviation tolerance. And not surprisingly, a lot of the size traits are related to that. That's not very interesting. But what is interesting from the floral perspective is anther length was the one floral trait that was associated with tolerance, in that a longer anther length was associated with more tolerance. Oh, and this tolerance measure is taken from the model residuals of the graph that I showed you a couple times ago, where you're regressing seed total in both the environments on each other. So just some overarching observations. In general, plants that were larger were always larger. They were larger under drought, they were under water. But we also found that those larger plants in general, but this wasn't a super strong pattern, exhibited higher percent reductions in their seed production under drought. We found drought stress resulted decrease for most side traits, so that isn't surprising. But the floral traits is a little bit more interesting. We found shorter corollas and styles and a decrease in nectar volume under the drought stress. And then anther length correlated with drought tolerance. That was the only floral trait that correlated with drought tolerance. And I'm still working on finishing analyzing this data. Still looking at a lot of the correlations. We have a lot of that done, but I didn't have time to include it. And looking at the relationship with these traits with expectation deviation tolerance. I'm also doing some work to look at multi-trait architecture and its relationship with tolerance, mainly through principal component analysis. And then we also have a lot of plasticity data. So we're also looking to is it the trait, or is it the trait's plasticity that is associated with tolerance? We have some root data too that we'll be looking at. So just wanted to acknowledge people that were involved with this project. I've already acknowledged Andres. Ben Blackman helped us in thinking about some of the traits we might want to include. Jared helped Lauren in figuring out how to measure nectar, and Oliver helped here at Central. So thank you very much. Questions?